My name is Charlotte. I work in Professor Titi's lab and I will be chairing today's webinar. Today's webinar is really interesting because it's a combined effort between an astrophysicist and a pathologist. And the title of the webinar is Astronomy Meets Pathology, an interdisciplinary effort to discover predictive biomarker signatures for immuno-oncology. So as you probably know, in advanced metastatic cancers, immunotherapy is often used, like even further than chemotherapy. And for that, it's really important to find predictive biomarkers, that is to actually see how the patient is responding to the treatment and then to make better decisions in the future. And so these two people, Dr. Janis Taube, they both work at Johns Hopkins Uni in America. Sorry, I don't actually know where it is. And so she specializes in the identification of potential biomarkers for the response to novel immunotherapies, and she's a medical doctor. And the other person is Dr. Alexander S Szalay, and he has a PhD in astrophysics. And he specializes in the statistical measures of the spatial distribution of galaxies and galaxy formation. So they both combined their knowledge to develop a technology called AstroPath. It's a platform that uses celestial object mapping algorithms to process the large amounts of data sets they get from immunofluorescence work, where they use immunofluorescence to get data from the tumor environment, but because of the large amount of data they have, that technology is needed. Yeah, and throughout the webinar, you can obviously ask questions like throughout the chat. I probably won't be able to answer them, but maybe we can find people from the audience to answer them. Yeah, and we can get started now.
Finally, thank you to Akoya Biosciences for sponsoring today's webinar. I'm now very pleased to introduce you to today's speakers. They will present consecutively, so I'll introduce both of them before handing over the microphone. Dr. Janis Taube is the Director of the Dermatopathology Division and Fellowship at Johns Hopkins University School of Medicine. Her research interests center on immune evasion by solid tumors and the identification of potential biomarkers of response to novel immunotherapies. Her ongoing work aims to further characterize the local tumor microenvironment with the goal of developing rational treatment combinations and improving patient selection algorithms. Dr. Alexander S Szalay is the Bloomberg Distinguished Professor of Physics and Astronomy and of Computer Science at Johns Hopkins University. He is a cosmologist working on the statistical measures of the spatial distribution of galaxies and of galaxy formation. He is the architect of the Science Archive of the Sloan Digital Skyserver and was project director of the National Science Foundation-funded National Virtual Observatory. A very warm welcome to you both, and Dr. Taube, I'll hand it over to you.
Thanks, Sean. Before we get started, I'd like to disclose our research funding. We do have a loan of vector imaging systems and receive reagents from Akoya Biosciences. Both Alex and I also serve as consultants for Akoya. An overview of today's talk, I'm going to first talk about a potential use case for multiplex IF and IHC and where we are in terms of validating assays across institutions. I'll then hand over to Dr. Szalay to talk about the unique facility that he's really spearheaded for our tissue imaging data using his experience in astronomy. And then I'll wrap up by showing one of the new candidate markers or phenotypes that we've identified using our AstroPath system. So in 2014, when we were looking at what I like to call the immunoarchitecture of tumors, we would look at serial sections from a single tumor block, and we would infer relationships. And so this is a melanoma deposit in a lymph node. You can see the dark arrows in the upper left-hand H and E show the melanoma deposit, and in the background lymph node, it's shown further on the left with the stars just above the germinal centers. And we'd be able to say, okay, you have some CD20 positive cells that are involving the tumor. There's certainly more CD3 positive cells, with a ratio of approximately 4:1 in terms of the CD8 to CD4s. And you can say that the T cells and even some of the B cells are in close proximity to PD-L1 and PD-L2 expression. And again, also infer that some of your cells are expressing PD-1 on the tumor-infiltrating lymphocytes. But with some of these multiplex technologies, it's now possible to put all of these stains on a single slide. You can also then split the composite image into the individual channels and see each of the stains individually as well. And those are shown here around the outside. But it's much easier now to identify true co-expression. Here you can see that the tumor cells shown in magenta are contributing a lot of the PD-L1 to the tumor microenvironment, for example. It's also a lot easier to study distance metrics. This is a process known as phenotyping, where each cell is identified. And so shown here are the FOXP3 positive T cells in green. That's each of the spots with a CD8 cell shown in red. And so quantification is facilitated, but also studying, what is the average distance between one cell type and another, from one of the centroids that's labeled to another centroid. And so now there are a number of biomarkers that are available and a number of different technologies for predicting response to immunotherapy. There is, your classic PD-L1 immunohistochemistry, which represents a standard surgical pathology workflow now. There's been a lot studied on gene expression profiling with a focus on interferon gamma. Tumor mutational burden, of course, has also gotten a lot of press. Now we're bringing in this new technology, multiplex IF and IHC. And the question is how does this stack up? In addition, a number of other investigators have used what I'll call multimodality, which is combining some of the other approaches. For example, PD-L1 IHC plus gene expression profiling, or PD-L1 IHC plus TMB, or even all three in some cases. So is there a distinct advantage for one or some of these technologies over the other? And to try to answer this, we did a meta-analysis of the biomarker papers that were out there predicting anti-PD-1 or anti-PD-L1 responses. We identified 135 records through PubMed search and almost another additional 100 through meeting abstracts. We specifically focused our PD-L1 efforts on Daily Med and those PD-L1 IHCs that were associated with approvals. We then excluded a bunch of these because they were not original investigations or didn't have a treated population. Some of the cohorts were too small. And we really focused on solid tumor types. In the end, we had 44 unique publications that had done 55 different analyses on one of these different biomarker groupings. This represented more than 10 tumor types and over 8,000 unique patient samples. What we did then was extract sensitivity and specificity values for each study for predicting response to immunotherapy. So shown here, responders in green, non-responders in red. This is an example of tumor mutation burden. We used whichever threshold had been used in that individual study. And then asked, what are the true positives, and calculated this 2x2 square for each. We then plotted the sensitivity and 1 minus the specificity for each study on a ROC curve. The ideal test is in this upper left-hand quadrant. This is an example of the studies that we looked at for PD-L1 immunohistochemistry. Here, the size of the spot represents the number of patients in the study. And so we then drew a summary ROC curve through the modality for each of the studies. So, for example, shown here, the blue line is the curve for PD-L1 immunohistochemistry. We drew the line in two ways. One that was weighted for the number of patients in each study, and one where each of the studies were treated equally, and that's shown on the right. Here's the blue line. The AUC for each of these is shown here, so 0.66 for both of them, 0.67 for the unweighted. TMB is shown in red, gene expression profiling, again, the interferon gamma gene signature in yellow. And you can see that the multiplex IHC and IF in each of these instances had significantly greater area under the curve than the other modalities that were studied. What if you actually combine the modalities? As I mentioned, a number of studies looked at whether it was TMB plus PD-L1 IHC, or here's one, TMB gene expression profiling and PD-L1. These studies started to approach the multiplex curve, but did not eclipse it. One of the important points is that the multiplex studies included in this meta-analysis actually only tested an average of 2 or 3 markers. And I think that really emphasizes the potential power of spatial resolution within the tumor microenvironment. Co-expression is another key variable which we think is very important in addition to spatial resolution, and having multiple markers, again, on a single slide also helps facilitate that parameter. There are certainly a number of technologies that claim the ability to test many more than 10 markers. And of course, the presumption is that we will continue to see an improvement in predictive value the more variables we test. So is it possible to standardize these assays and use them across institutions in clinical trials and perhaps ultimately in clinical care? To do that, we've been working with a number of other sites on testing an optimized 6-plex assay. This is shown here for lung cancer, and the panel includes cytokeratin, PD-1, PD-L1, CD8, CD68, and FOXP3. And visually, you can see across the 6 sites how similar that this staining appears. Our partners include academic institutions as well as pharma and Akoya themselves, and we tested reproducibility of the different markers both on tonsillar tissue as well as on tumor microarrays. Both our inter and our intrasite reproducibility for cell densities of each marker were on the order of 0.8 for our R-squared, so good reproducibility across all sites. We then wanted to look at some of the more complex parameters that I mentioned, co-expression, for example. And so here we're looking at percent PD-L1 expression within cytokeratin cells, as the dot plot shown on the left, and then within CD68 positive cells shown on the right. And again, our average concordance was above 0.8 for those parameters. We also wanted to look at the reproducibility of proximity assessments, specifically the PD-1 to PD-L1 assessments. And again, a robust measurement was obtained across all sites and was comparable, really indicating nice agreements in assay performance. One of the challenges in using these technologies is really the amount of data that is generated. So a lot of the early presentations and papers have really only assessed somewhere between 5 and 10 high power fields per tumor. And shown here is a representative lung cancer case that actually has over 1,000 fields when you look across it. I'll also remind you that the data that I just showed you from the multi-institutional study, which is the MITRE study, looked at tissue microarray spots, which are each on average only 4 high power fields. Why don't we go to more fields? It's really a time and data usage issue. So to map this entire tumor, as I mentioned, takes over 1,000 fields and over 300 gigabytes of disk space. And so when we expressed an interest in wanting to look and query whole slide data, I'm very grateful to some of my colleagues at Hopkins who pointed me to Dr. Alex Szalay, and he's helped us with our tissue imaging data using his lessons from astronomy.
So my journey in big data started with when Johns Hopkins joined the Sloan Digital Sky Survey. The Sloan Survey, SDSS, sometimes we like to call it jokingly the Cosmic Genome Project. It started roughly at the same time as the Human Genome Project with similar goals to map the whole northern sky, take high and detailed images of the northern sky, and then use this images to create an ultimate map of the galaxy distribution. And in the process, we worked on, so we finished the first incarnation of the project in 2008, and we collected over 5 trillion pixels of imaging data, and we built a 35 terabyte database in the end, and one of the products was a beautiful high-resolution 3-dimensional map of the galaxy distribution. What is important about this, that the whole idea of the project was to make the data immediately open and public and available for everyone. And our group was involved in building the database for the SDSS. And we built something called the Skyserver in collaboration with Jim Gray from Microsoft, who has been one of the world's most well-recognized computer scientists and database architects. And together we built a system on top of the database, which enabled us a visual navigation of all the spatial data, and people were able to interact with the data on many different granularities. And so over the first 16 years of the life of the database, people submitted several billion web hits. They typed in over 400 million SQL queries to extract detailed data sets and download them. It resulted in many thousands of refereed papers. And what most amazingly, we know that there are only about 15,000 professional astronomers in the world, so our community is much smaller than in life sciences. Yet there were millions of users with distinct IP addresses who actually not only just looked at images of the sky, but they actually tried to do something interesting with the data. So we saw the emergence of the internet scientists. And so there are lots of lessons we learned over these last, actually 28 years. So that there are strong parallels between medicine today and what astronomy was 25 years ago. So we see that stars and galaxies are like the cells in the pathology. They are both kind of the atoms of the respective science domains. So stars and galaxies are the atoms of the universe, whereas cells are the basic building blocks for pathology. And the way we acquire the data is through multicolor photometry, image segmentation, and a very strong notion of locality. And spatial relations are extremely important. So how the galaxies are clustered in the universe tells us a lot about the underlying dynamics about the dark matter. At the same time, and spatial searches are very relevant. And we actually have to store the outlines of the large structures. And there's a very similar challenge in pathology where we want to study the tumor microenvironment. So what are the lessons? So first of all, the statistical analysis and collaborations are much easier in a database where the data is kind of securely locked down than with hundreds of thousands of little pellet files scattered around disks. The other lesson was that we had to find a common processing level where the whole
Sorry, Luca, I think you're muted.
Lot more code. Essentially, we had to rewrite a lot of the fundamental image processing packages. We wrote over 1 million lines of code to actually get this done. And even moving many terabytes of data is hard. So we built this flexible browser, spatial browser on top of the database. Jim Gray was actually the person who built the first such MapQuest and Google Earth equivalent called TerraServer, which was the basis how we started out on the Skyserver project. And so here one can see actually in the database, we track even the pixels that belong to each galaxy, which was the region of attraction for each galaxy. These were the pixels where they contributed to the flux measured for that particular star or galaxy. And you can navigate through the whole data set without actually typing anything, just by clicking interactively and zooming and panning on the sky. So when we started to work together with Janis, we looked at how can we get on the path towards scalability. First of all, we have to take, of the order of 1,000 times more images. So we have a couple of slides where we have 2,000 to 3,000 high-powered fields, and altogether in our first clinical cohort, we had to scrape through 2 trillion pixels. That's about half of what the whole Sloan Digital Sky Survey has done in its first 18 years. That was just our first first 79 slides from a clinical cohort. So take dramatically more images. We also have to dramatically decrease the human labor. So we had to come up with the right protocols for the image processing and how the files should be laid out on the disk when they were acquired by the microscope, so that we can automate the whole workflow. Then how can we automate the tissue segmentation? This is a problem that we are still very actively working on. But we then have to build a spatial database that is scalable to many billions of cells. So currently we are roughly at around 100 million cells in our database. And eventually our goal is to integrate with genomics and medical records. So this shows roughly the workflow. So here we have the microscopes, and the data is collected on a server, and there is a quick preview and manual annotations are done over at the medical school. Then we have a 100 gig network called Hornet between the two campuses. And then the data comes to our data center, where we are actually doing additional processing, image corrections, and then load it into the database and then serve it up to the wider audience. So this was actually the first slide that we have imaged with this mosaicing technique. So the way we acquired the imaging data on the Sloan survey was to take overlapping images of the sky. And here the overlaps, let me show some more detailed pictures of these overlaps. You can see that there is roughly a 20% overlap between the adjacent slides. This is not an accident. So it turns out that this set of overlaps enabled us to design a fairly elaborate scheme. So the images overlap. So there are some parts of the tissue that are observed once. There are other parts which are observed twice or 4 times. You can see it on this image. So these are cells which have been observed once. Those are the blue, the deeper blue are cells which have been observed twice, and the orange ones have been observed 4 times. And these are independent pixels, independent part of the camera. And we can combine actually the uniquely observed areas and part of the doubly observed to create a seamless styling of the whole slide. At the same time, we can use the overlap regions to serve as quality assurance tests to determine intrinsically the uncertainties and errors in flux measurements and so on. And when we have done this, so this shows actually how this progression. So you can see the faint blue lines. These are the boundaries of the images which overlap with each other. Then this shows when we define the median lines between those overlaps, and they form a seamless styling. So it's like as if we were then cutting through with a recto knife, and then stitch it together, stitch the images together, and they form a uniquely seamless styling. This forms the statistical data set that we analyze, and this is statistically uniform. And so the rationale behind the overlaps is that they provide independent photons for the same cells in the overlap. These are repeated measurements, and we are able to determine uncertainties in individual cell fluxes. So if we have more overlaps, there's a trade-off here. So if we have more overlaps, we get a better signal to noise in these error measurements. But if we have too much overlap, in a sense, it's a waste of resources. Then we have to buy twice as much displays and so on. We can also measure in the overlap systematic errors in the image distortions, etc. And so this shows, the first image shows what happens in an overlap area with quadruple overlaps. And then I will magnify a little part of the top of the image. And so I don't know how well can you see on your screen, but there are little rainbows in the overlap areas where the complementary colors don't exactly overlap and don't cancel. This is the sign that there is a little bit of a pincushion distortion in the images. And by actually looking at this, we were able to fully correct and take out, so did an inverse ray tracing of the lens, and then corrected every one of the images so we get a subpixel alignment. This shows, and this shows actually how the trade-off works. So it turns out that the optimum point is in the amount of overlaps is 20%. So this is when kind of the two different costs of getting a better signal to noise and then still keeping the resources low is exactly balancing itself out. Okay, so when we saw this image warping due to the lens distortions, we took the overlapping images, and for every pair of the images, we did a registration. And basically, it turns out that there was a little bit of a jitter. This was a Vectra 3 microscope, and there was a little bit of a positioning jitter of the order of 3 to 6 pixels. And so this is the distribution of how much we have to move the pairs of images relative to each other so that the pixels exactly line up. And we consider these as if they were little springs that were stretched, and then created an overall solution for the system of the whole images, how we should tweak and move every image so that on the whole, the overall alignment error is minimal. And the scatter plot on the right hand is basically the alignment errors after we have made this correction. And this shows the probability distribution, the broad distributions, the broad wing distributions were basically the marginals of the scatter plot that I've shown before. And then the orange, narrow, sharp Gaussian in the middle is the after our image correction. So you can see that we reduce basically the positioning uncertainties by a factor of 5 to 7. It turns out that the Polaris, the new version of the Vectra microscope, is already much better. So basically, it performs as our sharp curve after the alignment. So we also saw that when in the overlapping areas, the intensities of the pixels didn't quite match. And we saw that there was a need to apply a flat fielding technique. In astronomy, we basically start every night with taking a flat field of the telescope, and we finish every night taking another flat field of the telescope. And we use this to correct every image. So this came naturally from astronomy. And when we smoothed the stack of 8,000 row flat field, 8,000 row images and smoothed it a little bit, we basically saw that there was quite a large scatter in the maxima and minima of the images. And then one of our postdocs devised a much more elaborate algorithm. And here there are different statistical measures as a function of the image layer in the 35 band images that we take. But essentially, the green is the RMS, the green contours are the RMS in the images, image intensities across the image. before the correction, and the yellow in the middle, the very thin line, is after our flat building correction. So you can see that's quite a big improvement. And then when we actually compare cells measured two different ways, one so without the image corrections and one after the image correction, you can see that in many of the different marker fluxes, there is the measurement errors are substantially reduced. So this is our processing workflow, which kind of puts together all these different steps, and then all the way goes to create data products that can be directly loaded into the database. And right now we have two pieces which require some manual intervention. One is when we create the annotations of the images, and the other one where we need to fine-tune the image segmentation algorithm somewhat. The rest of it is entirely automatic. And in the data, and basically every slide goes first into its own little database. So basically this is a slide DB load, and then this can be actually loaded in parallel. So we can throw a lot of servers and virtual machines into this loading, so this can be done quite fast in elapsed time, in real time. And then eventually all these are then merged into the master database, or the slide databases are going in essentially, all these sample databases go into a whole slide imaging database. And then all the common things about how the different chemical batches for the staining were done, how the calibrations, we calibrate actually, we have calibration slides in every batch included, and the clinical data all are contained in a core database whose data is then merged with the relevant slide data in the WSI database. So what is very important is the database contains a huge amount of spatial features. And these are represented as spatial polygons using GIS grammar. And we represent all the geometric outlines, the manual annotations, automatic tumor boundaries, membrane outlines for each cell, and the nucleus outline for each cell. So for 100 million cells, we have actually a little polygon for each. And then we have also distances. Each cell has its signed distance from the tumor boundary. So this shows, for example, the outlines. There's a good tissue and the tumor. Then this is a high resolution contours of the regions containing the high density of tumor cells. And this is how the whole tissue is covered with the outlines of the high powered fields. This is out of the database. And then we can take this polygon, so we can take the tumor boundary and grow this outside by 100 microns or grow inside 100 microns, and we can query the cells that are inside those buffer regions. So we can study the systematics of the tumor microenvironment as a function from the tumor boundary. Furthermore, we built actually a table called neighbors, which contains all for every cell, we computed all its spatial neighbors within about 50 microns. And this can be very useful. So with a few lines SQL query here, and using the geometries, I was able to compute the contact neighbors of every cell. So I could say that, okay, so for each cell, give me all the cells where the membranes are physically touching. And this is just a visualization of some of these cells, and this runs in about 5 minutes in the database. And we also, over the last few months, we have been working very hard to create the equivalent of the spatial browser for the sky, and we call this CellView. So the goal is to create visual tools for the whole slide image and with detailed spatial information, and give the users the ability to manipulate every aspect of the images and overlays, showed if for some cell types show only the nucleus, for others show only the membrane, and so on. And this is done integrated with the database. This is already in alpha testing, and the design is extremely scalable. So we build the whole thing so that we can eventually move it to the cloud and create an open and public atlas of tumor cells. And the database is already integrated with Jupyter and the Python, R, and AI backend, where we can very easily pull the data into all these different toolkits like TensorFlow, from the Skyserver, and essentially power users can perform machine learning and AI projects on the data. So the current status is that we are taking 1,000 times more images. We have taken currently about 200 slides, and this imaging quantity is roughly equivalent to all of the Sloan Digital Skysurvey imagery. And I cannot thank enough Acquia for the wonderful microscopes they have provided with this high throughput. It dramatically decreased the human labor, so our automated processing workflow is almost complete. We are converting everything to Python from MATLAB. And so Siyun Park and Ellen Yule are two experts in machine vision, and they have been working on deep learning for the cell recognition. And we are well on their way to create this flexible analytics environment where in the Skyserver, people can use and collaborate and share intermediate results while they are doing the analysis of the data. And so we have Python and R access to the databases directly, and each user can have their own database to create value-added products of, for example, cells which are expressed in certain expression markers. And so the CellView is the interactive spatial browser. So, and this is the Skyserver. You can try it out if you go to skyserver.org and try to play with it. And with this, I would like to hand it back to Janis, who will talk about how we have used the database for new candidate markers.
Hey, Janis, are you there?
Yes, I'm here. Thank you. And thanks, Alex. We had a couple of questions that we were very excited to ask in the database. And so I mentioned some of the advantages of multiplex technologies in terms of spatial arrangements and ability to study co-expression as well as tissue sparing. But these technologies have often been described as flow cytometry on tissue, and intensity is something that I always think of with flow cytometry, and we were very interested in querying whether we could reproducibly assess as intensity on a slide, and specifically PD-1 and PD-L1 that have been known to associate with different functional states in cells. So the first question we wanted to ask was, do PD-1 and PD-L1 expression intensities on individual cell subsets associate with clinical outcomes following anti-PD-1 therapy? And I'm going to focus on PD-1 specifically here. The other question that we had was really about the optimal TME sampling strategy. So I mentioned before that a lot of studies to date have looked at either 5 or 10 high-powered fields. They've also looked at them, depending on the study, sometimes in so-called hotspots where there's dense immune infiltrates, but also others have made a deliberate effort to do so-called representative sampling, where they're looking at both the core of the tumor and the edge. There's been a huge push also to move towards whole slide acquisition, and that's certainly something that Alex's Astronomy database has been able to facilitate. But we also didn't know if potentially acquiring a whole slide would almost be too much real estate. So now that we have the slide, we could look at optimal TME sampling strategies. And so Alex mentioned our first cohort. We had pretreatment specimens from 73 patients with advanced melanoma, and we stained them with a 6-plex assay. So PD-1, PD-L1, CD8, FOXP3, CD163, and our tumor marker, because of melanoma, we used a combination of SOX10 and S100. We had the tiled images across the entire TME, and that was again acquired in an unbiased fashion. We then ranked those tiles by CD8 cell density in decreasing order. That was a representation of a hotspot approach. But again, this is not user driven, right? This is done by the computer and therefore should be highly reproducible. We also assessed representative fields where there was a mixed selection between the inflamed and non-inflamed, as well as the central and peripheral areas of the tumor. And so if you do the hotspot sampling, this is just looking at CD8s. Shown here is an increasing number of high-powered fields across the bottom. And then we started the talk also by talking about the area under the curve, which is a global metric of sensitivity and specificity. So we looked at the AUC for an increasing number of hotspots for CD8 predicted response to anti-PD-1. And you can see that there's actually the most predictive value was somewhere around 20 high-powered fields, whereas if you did the so-called representative sampling, you never got the same AUC type inflection, nor were you able to achieve the same high levels of an AUC. And this was consistent when we looked across all of our markers, and specifically when we started to look at PD-1 expression intensity. So now I'm moving beyond just CD8 to look at some of the other markers in our panel and different cell types. So at the top, we're looking at all cells that are present, and we're splitting into tertiles here. We have PD-1 negative cells. Those are expressing PD-1 at low levels, at mid levels, and at high levels. And then we're looking at those same tertiles of PD-1 expression across cells that are CD8 positive, those that are FOXP3 positive, those that are actually positive for both FOXP3 and CD8, and those that are negative for FOXP3 and CD8. And we did a separate panel that showed the vast majority of these are CD4 cells, as you might expect. Lo and behold, the best biomarker, if you will, out of this set in terms of co-expression ended up being FOXP3 CD8 cells that expressed PD-1 at mid levels of intensity. And the best predictive value was seen when you selected an average of around 20 high-powered fields across the slide. And again, these fields were ranked based on CD8 density. So the whole slide was acquired. And it was important to acquire the whole slide. But then we used that information to improve our signal-to-noise ratio and therefore get a great discriminatory power using this specific cell type and co-expression. So what's important about this, there is optimal sampling strategy. It's a little bit of a Goldilocks thing. The 5 to 10 high-powered fields that we've all been using for the most part so far is probably an insufficient description of the TME. And yet at the same time, if you assess the entire TME, you run the potential of diluting your discriminatory signals between them. And what about this key phenotype? This was not a cell type that we set out to look for. There's certainly some literature out there. I know Liz Jaffe has published on it. There's also some literature from the GYN cancer groups about CD8 FOXP3 positive cells. They had initially been described as potentially CD8 suppressor cells, but it's thought now that they represent early effector T cells at the earliest stages after priming. We found that they represented on the order of 3% of all CD8 cells in the melanoma TME. And to actually support this purported function, we were able to use AstroPath to go back and map their location, and they are indeed immediately present at the tumor inflammatory stroma interface, suggesting cells that have just been primed. All of these biomarker approaches are certainly works in progress. And I know I mentioned TMB as a global metric, but certainly not all mutations are created equal. There are mutations in important genes, for example, STK11, which is really important in lung cancer resistance to immunotherapy. I focused on the gene expression profiling, meaning an interferon gamma gene signature, but there are, of course, thousands of genes, and those also can be spatially resolved. And innumerable markers to potentially explore in multiplex IHC and IF. And so I think we are at this point of information explosion, and the question is going to be how best to boil these down to an actionable biomarker which we can optimize and deploy in a clinical context. And so for us, we're hoping to use the lessons from astronomy to achieve that, taking the whole slide equivalent to the whole sky, our tumor microenvironment akin to the cosmic web, cells being the equivalent of galaxies, and after segmentation, then moving on to some of these spectral signatures. In conclusion, our early results indicate that multiplex assays are reproducible across institutions. Our guiding principle is really that the next generation of tissue-based biomarkers will be identified using large and well-curated data sets. And so to that end, we've developed a unique facility to produce petabytes of tissue imaging data. And as Alex mentioned, this number continues to increase. The melanoma projects that I showed you had 60,000 high-power fields with over 83 million distinct cells, but we have certainly already outstripped those numbers with the additional cohorts added.
Okay, that was the end of the webinar. So if you have any questions, you can just ask them. You can raise your hand or just write them in the chat. There's a comment from Mark. He says, I think the most important problem of this kind of approach would be the hard drive space. Probably something like this could be used focusing on the hotspots, which seem to give the best predictivity. What do you think? Yeah, so if anyone has something to say here. Yeah, well, you can think, and maybe someone can say something in the comments, but Professor Teti also has a question. I find the overlap picture alignment issue very intriguing, as it makes a lot of difference if it's done properly or not. Are students and fellows aware that it is not possible to play with scientific imaging in a non-professional way? Perhaps you can open the discussion on this aspect.
Charlotte, I raised my hands.
Okay. Sorry, I didn't see it. Okay.
No, it's just because I think that some discussion can be done just opening the microphone, because
Yes.
we are not expert, and so it's difficult to answer properly this question. The question that I think the issue that Marco raised brings me to another point that I would like to add, that is working on histological sections for this large-scale analysis. I don't think it's very easy, because sometimes the sections are not perfect for various reasons. It could be because the sample is not perfect, because the inclusion is not perfect, because the person who is working that make mistakes. So when you acquire the imaging and make all this elaboration, maybe you can measuring or investigating spots like the hotspots that were discussed and Marco was asking, that are not hotspots, but they are artifacts. So I think that this is an issue that needs to be taken into account. So maybe, I don't know if Marco has something to add, or if he can explain a little bit better his point.
Yeah, absolutely. That was also my, let's say, criticism about the work. So of course it would be more practical to focus on these so-called hotspots, but at the same time, who decides what is a hotspot. Maybe that could be defined in a more strict way. In the talk, they spoke about immune cells hotspots, but I don't know. Like it's I find it very hard to make this consistent among let's say all the medical institutions in the world. Would be very hard to implement in such a way, with having such a subjective way of defining some of the areas. I don't know if you agree on this point, Professor.
Yes, I agree that this type of investigations sometimes are really arbitrary, depending on your interest. So what you are looking for, and so this can change the results. And so I agree, but I would not be worried about the development of these techniques, because this is work in progress. So maybe in a few years from now, many artifacts will be solved, and the technique can be used also routinely.
Yes,
So
I think, I think so too, because I mean, every technology at the start might be a bit, obviously this would be quite exhausting to use this technique now, but I think that is the case for most techniques in the beginning. And like you said, if the problems were resolved, maybe it would be able to use as like a routine practice, yes.
Yeah. No, I absolutely agree, because with the trials and errors, the techniques will become perfect in a few years, probably. However, it's also important to understand that who is using the technique must be well prepared, because the artifacts are many and interpretations can be many. So I think that you should not trust only the machine, but you should also ask the proper question, make hypotheses, and validate your results with more classical method. Because with this very sophisticated and very sensitive methods, even nothing can appear as something. So it's and my other point was when he was discussing about the overlapping pictures that need to be aligned properly, and he showed that when they are not aligned, you see that there is no perfect overlapping of the signals. Again, this comment is for the students and the fellows that have no enough experience yet, and they have to know that they have to check everything very carefully, because it's very easy to do mistakes without being aware that you made a mistake. Because I'm sure, I mean, if I were a young person, I would be really enthusiastic of using this material. But my experience say that sometimes your eyes still see better than the machine. And if the two things do not match, you have to be careful and maybe ask yourself, where is the mistake? I don't know if I made it clear. So for this reason, I think that this could be a good point of discussion, and maybe some younger students can say something.
Antonio says, I agree that most of the time the problem is more related to the people and not to the technique. I also wanted to add, since she said this, she wants to use this to kind of guide the immunotherapy in cancer patients. So this means that this will also not be employed by scientists, but by doctors, because obviously, like, you cannot have like a scientist make a diagnosis for every single patient. So it will be a doctor who's not maybe trained in that field, actually.
Well, I think that you need a team. You need a team of people with different expertise. The final diagnosis and decision is taken by the doctor, no doubt. But what I see now is that sometimes the doctor do not have a hypothesis of diagnosis. But they start looking from using technique A, B, C, D, and then they get confused.
Yeah.
Instead, I think that it's still valid that according to the situation, you make a couple of 2-3 hypotheses and test these hypotheses without going random, because random it's very expensive. You waste a lot of time because it takes months before you get the diagnosis. Whereas maybe if you are more prepared scientifically, you can say, well, this symptom matched with this other symptom and with this other symptom, maybe the diagnosis could be this one. I think that, this is something that really changed a lot. So there is a lot of improvement in imaging because, for instance, they can discover very small tumors, identify very little changes that maybe are important. But if you are not able to use these methods properly, then you can provide the wrong diagnosis.
Yes.
I would like to ask, for instance, Michaela, you have an immunological background. Did it make sense what they were investigating?
Hi, can you hear me?
Yes.
Yeah, it makes sense. But what I'm thinking is, I'm completely agree with you. So when I was in the hospital where I study, we use cytoperimeter techniques, but for sure there is an equipe. So we have the doctors and also the researchers that ask questions together. And also I think that it is very important to decide the right test to make, because doing the routine tests randomly could be unuseful, as you mentioned. For example, if the patient use some drugs like, I don't know, anti-B cells, and you want to see the B phenotype of these patients, could be unuseful for sure. But if the doctor don't speak with the technicians, maybe sometimes happen that you make tests that is very unuseful for the diagnosis of these patients. So yes, I'm completely agree with you. So it depends patient by patient. Also, you have to think before what you are doing and then.
Yeah, and I think that in the team, not only you need the scientists and the doctors, but also technicians that know exactly how the method. This is another point. In the past, there were many technicians in the labs, at least in Italy, specialized in certain techniques. I think that in other countries, and probably Charlotte can confirm, also Katie in UK, the technicians are still very important. So each researcher has 1 or 2 or even 3 technicians rather than many students. Right, Charlotte? Is it like this?
Are you talking about in a science environment? Or?
In a science environment, yes.
I'm not sure about that actually, because I wouldn't even know what qualifications does a technician have?
Yeah, the technician is, for instance, in Italy, people who take the bachelor degree.
Yeah.
They can be technicians because they make the experiments, they evaluated the experiments under the control of the scientist.
Okay, because in Germany we have, it's an apprenticeship that you do. So there are people in the lab who are technicians, but they have not a university degree.
Yeah, sure, yes. But this happens in university, also in companies and so on. It's very typical. In Italy, this has changed. But I think that we should go back to having great technicians in our lab that needs to be very much specialized, not in routine techniques, not in everyday work, but on this sophisticated techniques. Of course, we are here and not in the parliament, so they will never listen to us. Because it is a great way also to find jobs for people who do not want to become scientists, but they like to to apply techniques of any kind. In the clinical, this is not the case because there are many technicians there, and
Okay.
so, but
Yeah.
not in Italy. In other countries, as I told you, is different.
Yeah, in fact, if I can add, when I was there in Rome, in this hospital, we have a lot of facilities. So we have people that are technicians that are able to manage microscope or some other instruments that have to be managed very carefully. So they have to be there for all the person in the lab. So I think that it will be very useful. So you are pretty sure that the experiment will be done in a right way because you have these people that are specialized in this. So for sure, it could be a very important thing.
But Michela, it is also the other way around. Even if you don't use your personally that specific technique, you have to know how it works.
Yeah, for sure. Yeah, for
Otherwise,
sure. Otherwise,
if there are, because also technicians make mistakes, and then you will not realize that then.
Yeah, for sure.
So
but
it's a reciprocal, interaction.
Yes, for sure. But it's useful because you can stay there for sure, and also learn about from this person, from these people. Yeah,
Sure.
how to do it, how to manage this instrument carefully and in the right way, so without any mistakes. So and you can also break this machine. So you have to be careful with this.
We have a long list of machines that are broken. Okay. So Charlotte, there is an interesting question from Marco. If there is any future medical doctors in the audience that want to comment, don't be shy. Either they are shy or they do not have doctors. So this is something that we should consider, trying to attract more doctors in the audience. Okay, maybe you can close the...
Yes, okay. I think if there are not more questions, then I think we can close for today. But I thought it was really interesting, and yeah, I enjoyed it.
Okay, so thank you.
Bye-bye.
See you next Thursday. Bye-bye.
Thank you. Bye-bye.